Good evening, ma'am. Hello, ma'am. Good evening, ma'am.
Good evening, sir.
Good evening, ma'am. Good evening, Ajay.
Good evening, sir.
Good evening, Kana. Good evening.
Sir, you can start, sir.
Yes, ma'am. Thank you. Thank you so much. Good evening, all of you. You are all are warm welcome to the another session of the biological classes.
Good evening,
Dos-
sir.
Good evening, Pa. Good evening. So today what we are going to discuss, already yesterday we saw it. Some important terms related to immunity. Okay, already we saw it. What is by immunity? What is by immunology? What is by antigen? What is by antibody? That things you must note it on. What are the terms, important terms, which is related to the topic. Okay, see here, the immunity, the resistance of the body against a pathogen or disease is called as immunity. Resistance of the body against, okay, we are having resistance powers naturally or artificially, okay, and that condition is called as, what is that? Immunity. We are having so many immunological system, otherwise called as immunological organ system, isn't it? They are all produced resistant substances, okay, that fight against many diseases is called as immunity. Second one is antigen or agglutinogen. Already we discussed it. It is nothing but the proteinaceous substances which stimulates production of antibodies is called as antigen. Okay, they may ask the question, antigen is also one of the proteinous substances which is produced by when the foreign particles enter or virus enters or alien species enter in our body. During that time, automatically the protein substances produced, okay, that protein substances produce antibodies. That substances is called as, which substance, which one is produced responsible for the production of the, which one, antibody. That substances is called as antigen. Okay, so go to the next one is, say, antibody. Already we discussed it. It is a complex glycoprotein. So many times they were asked the questions. The antibody is one of the complex glycoprotein or simple protein or globulin. Like that, they may ask the questions. Complex glycoprotein secreted by B lymphocytes. Imagine antibodies are secreted by the one of the glycoprotein molecule secreted by beta lymphocytes in response to an antigen. It is called as agglutinin. Agglutinin is nothing but the physiological process when a corresponding antigen react with the antibody. It undergoes the clumps-like structure, just like a blood clotting mechanism. That condition is called as agglutinin. Okay. Then next one is antiserum. Already yesterday we talked about these things. Okay, regarding serum, what is my antiserums? Serum of the any animals which contain the antibody for a specific antigen is called as antiserum. Serum of any animals which contain the antibody for a specific antigen is called as antiserum. It will be obtained, it is from horse or rabbit or monkey or whatever it may be, rabbits also. Okay, the antiserum also isolated. Take an examples of, how will you identify the blood grouping? How will you identify a blood group?
So,
Yeah. One of the.
so we can identify the blood group by the antibody which it contains, sir.
Okay.
So it can be either antibody A, sir, or antibody B, sir, or
No.
A, B, sir, or no antibody, sir, which is O.
So, antibody or antiserum? Antibody or antiserum, Ajay?
So antiserum, sir.
Right only. Okay, good. So antiserum is one of the, which one, kids. About the, how will you identify the positive and negative blood group? So you are having positive, isn't it? Blood group. A positive, B positive. What about your blood group?
Sir,
Ajay.
RH factor.
RH factor. So RH factor denoted or decided whether positive or negative, isn't it? There is also one of the antiserum isolated by RH factor. RH denoted as rhesus monkey, isn't it? You must know your blood group whether positive or negative. Okay. Suppose if it is negative, we will study that things also important for the which one, you know, physiological functions. We will discuss later. Okay, anti-serum. We go to the next one. The venom. Venom already in our venom is nothing but the poisonous, otherwise called as toxic substances secreted by the snake and also some insects. Some of the insects also. Take an examples of even the sea. There is one of the octopus or jellyfish. Okay, jellyfish. Sorry, jellyfish is also there. Jellyfish is also, it has a venom or poisonous substances. Take an examples of scorpion. Okay, and also other. Okay, reptiles. Some of the reptiles they are having the poisonous. Particularly, tell us a snake. It is very dangerous one. But at the same time, the venom is also modified and also some alternation is also done by the scientists and also used as a medicinal purpose. Okay, this is also another one. Agglutination, already we discussed it. When the antigen and the antibody reaction is called as agglutination. When the exact matching of the antigen react with the antibody, during that result, what happened? You know, the clumps together. Okay, otherwise called as, what is that? That is clatting together. That means bond between the antigen and antibody. So as a result, the clumps, it happened. We can see that. We can see. Okay, during the antigen antibody reactions. That condition is called as agglutination. When you are going to see the, which one, you know, blood grouping, when you are going to analyze the blood grouping A, B, O, whether positive or negative. When you are adding the anti-serum, what happened, you know, that is, what is that? Antibody react with in your antigen, corresponding antigens. As a result, the agglutination happened. Okay, that is called as, what is that? Agglutination. That process is called as agglutination, otherwise called as clumping, otherwise called as antigen antibody reaction happen. As a result, you can see the clumps in the slide. Okay, in the medical laboratory, DMLT, okay, deployment medical lab technician. When you are going to analyze the blood group, you can see that slide, it may be clumps in manner. Okay, so that antigen antibody reaction is the study of the antigen antibody reaction is called as serology. Note it down. This is very, very important. Sometimes they may ask the question. Okay, toxoid, yeah, bacterial exotoxin. So exotoxin present in it at the same time. Which one is also present? Endotoxin is also present. Exotoxin means outside. Endotoxin within the cell, which is detoxicated by special procedures to allow its safe use in immunization against the diseases. Suppose any one of the person bite by the snakes, automatically the venom, the poisons enter your body. Okay, enter some person. As a result, what happened? You have to remove the poison from the particular person or person. How will you? There is a special procedure for the detoxification. Okay, to allow the safe use of immunization against the diseases, here against the poison. You have to tell. So go to the next slide. And interleukin. So what is the meaning of interleukin? It is also one of the byproduct or genetically engineering product. Okay, it is also one of the protein molecules. See here, it is a protein substances which stimulate the growth and activate certain kinds of WBC that are involved in the immune response system, also act as a secondary messenger that activate the immunes. Interleukin is also act as a secondary messenger. It activates the immunological system. The immune system is also activated by which one, you know, interleukin. Not only that, and also the WBC also what is that? Motivated, activated by certain kinds of WBCs. Okay, see, there are so many examples as given. So example, they may be divided. Subdivision is also there. Interleukin is also discovered in the laboratory. They may be divided according to the protein nature. They may be divided in interleukin 1, interleukin 2, interleukin 3, interleukin 4, interleukin 5. So these are all how they are differentiated, because depends upon the protein amino acid sequences, because it is also one of the protein substances. Okay, they may be vary. And then we'll pass on to the last one is called as immunity. So already we discussed what is by immunity. It is divided into two types according to the nature. One is called as congenital immunity or innate immunity or non-specific immunity. These are the three names for the one. Congenital immunity, or otherwise called as innate immunity, or non-specific immunity. This is the first one. Second one is acquired immunity or adaptive immunity or specific immunity. Here non-specific, here specific. Here innate, here adaptive. Here congenital, here acquired. These are the two types. They may ask the question whether they may ask the question non-specific, okay, instead of congenital, or innate instead of congenital, or innate instead of non-specific. Like that, they may ask the question. They may be shuffled. Okay, that is a confused in this status, isn't it? So you have to clear about it. Congenital, innate, non-specific, all are same. Acquired immunity, adaptive immunity, a specific immunity. These are all our same name, isn't it? Okay, see there. Immunity divided into two things, innate and acquired. Innate is the one of the inborn genetic factors, inborn from birth. Okay, the acquired immunity divided into two. One is called as active and another one is called as passive. Okay, the active antibodies, that is active, acquired immunity also for the event, active and passive immunity. The active immunity is nothing but the own antibodies present in our body. The passive is nothing but the ready-made antibodies. When you are going to take the hospital, you are getting the, what is the antibodies? That antibodies commercially prepared. Okay, that is called as passive. Further, active immunity is divided into two types, natural and artificial. Active immunity is also further divided into natural and artificial. Same aspects, the passive also divided into natural and artificial. Once again, repeat for you. Immunity two is there, innate and acute. Otherwise called as specific and non-specific, congenital. Okay, that like that. The acute immunity divided into two types, active and passive. Active divided into two, natural exposure to infectious agent and artificial immunization. Likewise, here also the passive also divided into two, natural maternal antibody and artificial antibody from the other source. These are the two types. Okay, we'll see one by one. See, the congenital immunes or innate immunity or non-specific immunity, it is started from the birth. It is also present by birth. It is also inherited from where, in your parent. The most animals and the forms natural defense system, which is always available to protect the body against various type of foreign agents or pathogens. It is present in our body. Okay, such type of immunity is called as congenital immunity, non-specific immunity, innate immunity. It is also called inborn immunity. That is very, very important. Congenital immunity is otherwise called as which one, inborn immunity. This is a non-specific type of defense. Okay, this is, we cannot able to specify. It forms of the first line of defense of the body, and it is made up of the following barriers. This is very, very important. So many times they were asked the question. First lines of defense is also called as which one? Congenital immunity. It's otherwise called as innate immunity, otherwise called as non-specific immunity. Again, it's called as inborn immunity. The inborn immunity is also called as first line of defense of the body. They are having so many barriers. Without barriers, there is no possibility of congenital immunity. So what are the barriers? We will see one by one. See here, the physical barrier. Physical, your body having the physical barriers also. Some of the organs, they do not allow foreign agents and pathogens to enter the body because you are having the skin. Okay, the skin also having the holes, isn't it? Sweating glands, and also there is a minute pore. At the same time, and also the eyelid, okay, lacrimal glands also present, in mucous membrane in nose. Okay, throat also, urinary tracts also having the mucous membrane. The following parts constitute physical barriers. First one is skin. The outermost layer, keratin of the skin is dead, stratum corneum. So the bacteria does not grow or enter it. the skin we are having a, which one, dead cells. Suppose a person having a, which one, dark in color. Okay, that is called as keratin. Black in color, like that, you may imagine. Suppose one person have which one, white in color. It will be easily affected by these diseases, white person. Okay, easily affected because they do not have the immunity powers. Less immunity he has or she has compared to those are having more keratins in their skins. Okay, so the bacteria do not allow. The pH of the skin destroy the bacteria because during the sweating conditions, what happens? It emits the sweats, isn't it? It has a own pH because it is alkaline pH, because it's also sodium chloride. Already you know very about sodium chloride, also excreted in the form of water, and also it deposits on your skin. Okay, that's why we are bathing. When you are roaming here and there, you will get, or when you are going to play, that time what will happen, automatically the skin emit the water droplets in the form of sweat. Okay, it is a, what is that, salty taste. Okay, and also the sebum of the skin also has antibacterial action. The sebum is of skin also has a antibacterial activity. So this is the very important thing. This is also comes under the heading known as physical barrier. Okay, physical barrier. What is the next one? See, the physical barriers: skin, mucous membrane, cilia, body temperature, and pH. These are the physical membranes. So these are the immunities: the innate immunity, adaptive immunity. Innate immunity, what are the things have been here? See here, all the macrophages. Macrophages, what happened, see, it will be prevent the pathogen. It will not enter inside. The first, the skin is there, center the blood is there, then inside what are the adaptive immunity cells also protect the pathogens. So that's why we are healthy. Otherwise, we will fall into ills. That is very, very important. See. So neutrophil, basophil, eosinophil, macrophages, and these are all our blood components, isn't it? Blood cells, WBCs. And mast cells also, dendrite cells also, natural killer cells. These are participate in the innate immunity. Adaptive immunity, T helper cell, cytotoxic T cell, native B cells, and plasma cell. These all are involved in the adaptive immunity. So the microorganism or pathogen, it will not enter into the body. These are the things. Okay, the physical barrier only we studied, isn't it? Go to the next slide. A mucosal surface, already told you. Mucosal surface, mucous coating of the epithelium of lining of respiratory, gastrointestinal, and is a urogenital tract also help in the trapping of microbes enter our body. Take an example of nose. There is a mucous membrane present in the nose. Okay, and also has small hairs also. It trap the microorganism penetration or entering our body. Gastrointestinal tract also, it has a mucous membrane. Already we studied. And urogenital, okay, urinary genital, already in our level. Okay, it has a mucosal surface. They will not allow the microorganism easily attract the microorganism on their surface. Mucous membrane, just like a semi-solid structure, gel-like structures. Okay, the mucosa contain mucosa cells and cilia. The mucosa entrap the microorganism, and cilia propel the microbes. Skin membranes and chemical barrier form the first line of defense. These are all the first lines of defense. Two things we studied here. There are so many things have been reported already. We saw it, this diagram: the skin, mucous membrane, cilia, body temperature, pH. When the pH is increases, the microorganism, it will not survive. Body temperature increases, some of the microorganism, it will be killed. Cilia is nothing but the minute hair-like structures. It cannot able to allow the microorganism. Okay, see these are the things. Instead of cilia, we are having which one? Hair, nose or skin or whatever it may be. Okay, so this is the other things. See, and physical barrier. See, physiological barrier is also involved. Okay, many physiological functions of the body make unfavorable environment for the growth of microorganism. As a result, fever, pH of the body, secretion, and cellular barrier, these are the physiological barriers. So what is already we studied? Which one is the physical barrier, a physiological barrier? You must know that. Okay. See, these are the essential: fever and pH of the body and secretion, cellular barriers also involved. See, the first one is fever. Increase the temperature of the body, they inhibit the growth of the microbes. Already I told you. Okay, suppose the temperature increases, inhibit the growth of the microbes. Here they may arise the question, sir, suppose if we fell into or fall into the diseases, any one of the diseases, the temperature will be increased, means what happened? We may bath in hot water, microorganisms completely removed. Yes, outer surface only. When you are drinking the hot water, what happened? Inside your internal, what are the stomachs? Some of the microorganisms trap or adhere on the surface of the mucous membrane. It will be completely eroded, and it will enter into the stomach. Stomach, it has a HCl, completely digest the microbes. pH of the body, it is also another important. Acidic pH of the various part of the body, such as oral cavity, stomach. The spelling is correct or not? Wrong. Okay. Inhibit the growth of the microorganism. This is also very, very important things. Early morning, without brush, think about our oral cavity. There are so many numerous microorganisms present in it. That's why we are brushing. Okay. So that is another thing. It is also one of the pH of the body. And secretions, secretion already you know, but secretion is nothing but the enzyme secretion. Secretion bodies such as eyes, lacrimal gland, sebaceous gland, which contain lysozyme enzyme. Already we discussed lysozyme is nothing but one of the cell-bound organelle. It is used for the, which one, you know, phagocytosis, otherwise called as a cell-lating process, and helping the cells. The lysozyme secrete the enzyme is called as lysozyme. Okay, it has antibacterial properties. That means, what is that meaning? When it foreign particles such as bacteria enter, automatically the lysosome activate. As a result, lysozyme enzyme secreted, it act upon the bacteria, completely lyses the bacteria. Okay, that's why it is called as suicidal bact. Itself also, what is that? Degraded. Okay. See, the HCl is the gastric juice, has a germicidal properties. Already I told you, know, HCl gastric juices contain so many enzymes, peptidase, okay, trypsin, lipase, and carboxypeptidase. So many enzymes are. Addition with the HCl is also present. Okay, the HCl. What will be the pH of the HCl? Gastric juice contain HCl. The HCl, it is a which pH?
So acidic pH, sir.
So you have to mention exactly which pH. Which
Sir, I think it is 3, sir.
No,
Sir,
slightly
2.45.
2.5 is a correct one. Exactly correct.
Yes, sir.
2.5 is a correct one. Okay, good. Good, Ajay. Super. So it's a gastric juice, has a germicidal properties. And next one is bile inhibit the growth of the microbes. Biles. What is the main use of the biles? Secretions. Bile secreted by the gallbladder, isn't it? The biles also inhibit the growth of the microbes. Sermon or ear wax. Ear wax, ears also having some wax, trap the dust particles. Okay, these are all comes under the headings physiological factors. What is the main use of biles, my dears? Biles.
Sir.
Yeah. Go ahead.
Sir, bile, sir, it is used in the stomach to bring down the pH of these stomach acid, sir.
No, okay.
Sir, to make that food which is acidic, since it is mixed with the acid in the stomach, sir, the food will be food will initially be acidic, sir. This bile will make the food alkaline, sir.
Okay, good. But one more is there. It is very, very important. There are two types of bile pigment is there. Will you please? What are the two types of pigments? Dhanalakshmi, Dhanusri, Ritika. Will you please? Anybody else? Bile pigment, two pigment is there. Okay, anyhow. So bile also secrete two big pigments. One is called as a biliverdin, bilirubin. These two are involved in the fat digestion process, fat metabolism or fat digestion process. Okay, just note it down. Okay, cerumen are also called as air waxes, traps the dust particle. It will not allow the microorganisms in yes. And then third one is called a cellular barrier. This is also very, very important. Certain types of leukocytes, example WBCs of our bodies, such as polymarbonuclear nucleosides. This is also so many times they were asked in the expansion aspect. PMNL, polymarbonuclear leukocytes, particularly neutrophils, monocytes. These are the two things. Okay, I will show you next when you are going to study the hematology. I will show the structures or blood components. And the natural killers are also lymphocytes in the blood, as well as the macrophages and tissues can phagocytes and they destroy the microbes. This is the cellular barrier. Particularly cellular means cell involved for the which one eradication of microorganisms. Some of the cells, what are the cells? Taken examples, neutrophils. It acts as a antibacterial activity. Eosinophils and basophils, like that, each one it has a specific functions. Removal and eradication of the microorganism. It will not allow the microorganism, and also it produces the pathogen. Antigens. Okay, see that one. They act as a soldier. Here the spelling mistake is of defense and also scavenger of the bodies. This is very, very important. Soldier. Which one is act as a police force? Our body, no. WBC also act as a police force. Okay, because it will not allow the microorganism into the body. That is the reason. They come out of the blood capillaries through a diapendices and engulf and also digest most of the microorganisms. This is called as a process. That is called as which one? Cell eating process. Engulf, completely digested with the help of the enzymes. Okay, and the complex molecules of the foreign particle completely degraded into simple molecules, whether carbohydrate, protein, lipid, whatever it may be split into carbon, hydrogen, oxygen, hydrogen, sulfur, like that. After that, it will absorb the nutrition substances. And finally, what are the waste is there, it will be eliminated in the other side. That is called as engulfing, complete digestions. Okay, so go to the next one. And cytokinin barriers. The fourth one is also there. The virus infected cell. Imagine, this is very, very important. The virus infected cell secrete the protein called interferons. This is also commercially prepared in the lab when you are going to study the genetic engineering, biotechnology. Okay, humulin, interferons. Okay, that may be indicated as IN, interferons, IF. Okay, antiviral protein. Imagine, interferon is also called as antiviral protein made up of 270 amino acids secreted by various infected cells. Which infected? Virus infected cells. That means when the virus enter in your body, automatically virus infected cell produce the protein molecule. That protein molecule is named as which one? Cytokine. Otherwise called as interferons. The interferons is called as also so many antiviral protein molecules. The interferons is also called as antiviral protein molecules. It fights against many virus diseases. It has a 270 amino acids. Okay, so this is very, very important. So the AIDS patients also, they will provide antiviral protein molecules, otherwise called as interferons. It's also very depends upon their molecular structures and also amino acid sequences. See that image already we discussed these things present in your body. Okay, immunity, acquired, specific, activate, and passive. Active, natural, and also artificial. Passive also divided into two. Already we saw it. These things also, isn't it? Same things also given here. Same one is also. See, active, passive, natural, artificial. This one is also natural. This one is also artificial. So already we discussed these things. Now we'll pass on to the one by one. Acute immunity, specific. The acute immunity is a pathogen specific. Okay, acute immunity is also called as pathogenic specificity. It is acquired after the birth. Before we studied the which one before birth or born, after the birth. So due to the previous infection or inoculation of the antiserum, what will happen? It occurs only in the vertebrate only. Supplement proteins protections provided by innate immunity. It is characterized by memory. This immunity recognize and selectively eliminate the pathogen. It has 4 unique features. Once again, repeat for you. What is my acute immunity? Just imagine what is my immunological memory. Please, what is my immunological memory? What's your memory?
So,
Yeah.
so immunological memory is when our body remembers a particular infection, sir, and then whenever that infection comes back, sir, the body's immunity is automatically activated to fight against that infection, sir.
Very good, very good. Exactly correct. Once the microorganism enter, automatically the antigen is produced in your body. Okay, antigen against antibodies also produced, isn't it? Second time it entered the same microorganisms. What happened? Automatically it is analyzed by the immunological system and fight against the particular second invaded microorganism. Second time. That is correct. Good. So this is called as immunological memory that is induced by the acute immunity. Okay. Any one of the microorganisms, any one of the antibiotics, they will not directly kill the pathogens. They will initiate the, what is that? Cell immunity or natural killer cell or lymphocyte production or bone marrow lymphocyte, B lymphocyte, T lymphocyte production cells. Only they may initiate. They will not kill any pathogen. Just note it down. This is very, very important things. Okay. See what are the 4 unique? See, specificity. The acute immunity is a specific for specific microorganism. Each one, we will not use for the typhoid vaccine for which one, cholera, isn't it? Each one it has a specific vaccination. That is the reason. Immunity. It is the ability to differentiate various foreign molecules. Okay, the specificity, cholera and also typhoid, mumps, measles, or whatever it may, cancer. Likewise, and COVID also. Different, what is that? Pathogen is also, each pathogen, it has a different, what is that? Resistant ability. Okay, capacity. Okay, that is called a specificity, is the first one. And then second was diversity. This system recognizes the vast variety of microorganisms. This is another one, vast variety of microorganisms. Suppose if we are introduced a particular vaccine or any one of the immunity power, it will not activate on the another one. So we can come to the conclusion. So that is not for the, which that is not used for the first one or first vaccine, like that. We may come to the conclusion. Resting vast variety of microorganisms we can recognize, which is not activated, which is not eliminated or eradicated this type of vaccination. Take an example, COVID shield, okay, and also Pfizer. So many, so it will not act upon it. It only partial, what is that? Prevention only, like that. And third one is discrimination between the self and also non-self. It can recognize self body of tissues and non-self foreign tissues and respond accordingly. This is the third one. Okay. And then fourth one, see the memory. Already we discussed the memory. When a pathogen enter the body take longer time to recognize and respond to it. This is called as primary immune response. But the memory of this encounter remains in the immune system. So once the foreign particles enter in your body, okay, the system is analyzed and also produced, which one, antibodies. Okay, and also survive for many years. For example, once you affected by the smallpox, it will not enter again, isn't it? Because the presence of immunological memory. Okay, see that. And acute immunity is carried out by the 2 special type of lymphocytes. Already we studied that things. There are 2 types of lymphocytes. There, one is called as B lymphocytes, another one is called as T lymphocytes. B lymphocyte is nothing but the bone marrow produced lymphocyte. What will be the main one? This is very important one. B lymphocyte is the very important and fight against many diseases. The B lymphocytes produce a group of protein in response to the pathogen into the blood to fight with them, called antibody. The B lymphocytes, bone marrow lymphocytes. Okay. T lymphocyte, thymus gland lymphocyte. Here, the thymus lymphocytes, otherwise called as T lymphocytes, they do not produce the antibody, but help the B cells to produce them. The T cells help for the which one, B cell. It induces the production of which one, antibody. Particularly which one, antibody, the B lymphocytes. Lymphocytes also act as a antibody, antiviral. Okay, so many things is there. Just imagine those who are suffering HIV, the lymphocytes, otherwise particularly tell us the monocytes completely drop. Okay, the normal level into, okay, it will be less than that of the normal level. We can find out. Okay, see that. So these are the two cells, B cells and the T cells. The B cell, it has the, okay, it attack invader outside the cells. Okay, invader outside the cells, but at the same time, T cell, it attacks invader inside the cells. These are the two things. B cells, T cell, both are fight against many diseases. It is produce the antibody, but it will not produce the antibody. It will help for the production of antibody for the B cells. Okay, each are the mutual what is that? Understanding, like that. Okay, see that one. The types of acute immunity, already we discussed. These are the barriers. These are the cells. Okay, what are the type of acute immunity? Already we discussed. Acute immunity, resistance developed by the individual as a result and antigen stimulus. This is the acute immunity. This is divided into two types. One is called as natural one and artificial one. Natural means what is the result from the clinical or inapparent infection by microorganism. Clinically, naturally. Okay. What about artificial? Resistance induced by vaccine. This is the natural one, because in our body having the which one, immune system, they activate upon the particular microorganism clinically. Okay. At the same time, the artificial means man-made. The resistance induced by vaccine. The COVID-shield is one of the artificial vaccine. Okay, we are giving the vaccine to the particular person. What happening? It may induce the, this is artificial vaccination, induce the antibody production within the host. So this is the things, artificial and natural. See, the acute immunity, active and passive. Active also divided into natural and artificial. Passive also divided into natural and artificial. Already we discussed. That things also again it comes. See the active, natural, artificial. And then passive also divided into natural and artificial. So first we discussed natural, isn't it? See the types of natural and artificial. Okay, here see that passive immunity. It is received passively by host without participation of contribution from the host immune system. Okay, the immune system is not participating in this category, passive immunity. That means it may be ready-made antibodies, which is also injected from the outside. Immunological memory is absent. In passive immunity, the immunological memory is not utilized here, because already you know, but natural immunity only, what is that? The immunological memory is activated. But passive immunity, what happened? We are giving the antibodies from the outside sources, mean the immunological memory is not useful for that one. Particularly here, what happened? No, there is no activation of immunological memory. So the immunological memory is absent here. And the ready-made antibodies are given. It induces the immunosuppressive individual. This is called as passive immunity. Active, natural, and artificial. Natural also already in our the activity is from the outside sources. It is called as passive immunity. See the passive also natural and artificial. See here the natural, passive, natural. The resistant passively transfer from the mother to baby. This is called as natural, innate. Mother milk gives passive immunity to a newborn child by colostrum. First mother milk. Note it down. This is very, very, very important. Colostrum is nothing but the one of the yellow colored fluid, pale yellow colored colostrum. Okay, also comes. It has a many what is that? Antigenic property, resistant property. So many immunoglobulin, particularly immunoglobulin A is very high in nature in this category. Natural and artificial. Passive natural means it comes from which one? Naturally, the mother milks contain. It is the mother milk. First milk is called as what's that? Colostrum. pale yellow color. Immunoglobulin A is very high in nature. Artificial means, you know, the resistance passively transferred to the recipient by administration of the antibodies. Example, human immunological administration. What are the human immunological administration? There may be anti-tetanus serum, ATS, anti-rabies serum, ARS, anti-diphtheria serum. These are all what is that? Commercially prepared from the laboratory aspect or commercial prepared. These antibodies, when we are giving, what happened on a particular specification. See here, the tetanus, we will not give for rabies ATS. We will not give for ARS ADS. Okay, you have to give the particular specific antigen or antibody react only. Then only the disease will be curable. Otherwise, it is not curable. Suppose we are giving opposite manner, what will happen? It will not cure. That is the reasons. Okay. So these are all comes under the getting known as artificial immune system. See here, innate, specific, intact skin, ciliary, these are all already studied. Non-specific, eosinophil, neutrophil, and natural killer, dendrite cell, macrophages, mastocysts, these are all non-specific. This is called a specific, this is called as non-specific. Okay. And then see, adaptive immunity, active immunity, natural vaccination. Natural means already in our, you are having the mucous membrane, isn't it? Artificial means vaccination is nothing but we are giving the outer source. Passive also same. Passive also having the antibodies. Maternal, when we are giving the mother milks, what happened? It's also fight against many diseases. But at the same time, immunoglobulin treatment also, we are giving the another part. Also. Both are same, but here comes from the passive active immunity. This one is called as passive immunity. Okay, see. The two types of active immune system, CMIS, cell mediated immune system or cellular immunity. Here the cell is important. Okay, cell is also participate so many immunity and also induce the immune system. That's why cellular immunity. The cell is participate the production of the what is one immunological activities there. That is the reason for cellular immunity. This immune system is based on the T cells. 60 to 70% of the T cells activated in this area. CMIS. So many times they were asked the which one, you know, the mnemonics word expansion. Okay, there are 5 types of cells. Okay, what are the cells? Here the helper cell, killer cells, killer T cells, and also cytotoxic cells, and memory cells, suppressive cells, antigen presenting cells. These are the five cells also present under CMIS. Okay, the CMIS comes under the active immune system. Okay, active immune system. See here, one by one. So helper cells, killer cells, memory cells, suppressive T cells, antigen presenting cells. Isn't it? These are the 5 cells we are going to study. These are the 5 cells or participate in the cell mediated immune response. Okay, see here. So this is the infected cell. Take an example. This one is called as infected cells. Okay, what happened is a major histocompatibility. Antigens also it has, and also major histocompatibility, and memories also it's bind with the corresponding antibody. That is also otherwise called as cytokine is also participate. When it bind with the memory cell, automatically what happened? The cytokine is also produced with the help of the T helper cells. There is a receptor. The receptor easily bind with the which one in a infected cells. See here, the killer cells, helper T cells, and also killer cells. Finally, what happened? You know, the during the mitosis, the which the memory T cells produce more number of which one, you know, memory T cells. It act upon the which one, you know, cytokine. There is an active spaces here. Okay, the active spaces here. It act upon the particular which one, you know, cytokine, the T killer cells. As a result, the cells are finally dead. It's also ruptured by the cells. So cell is activated, also kill the another cells. That's why it is called as cell mediated. In between the two cells, which one is that? This one is a macrophage or infected cells. Okay, the infected cells also what may happen? Dead. How it will be dead? With the presence of the mediated cells. What are the mediated cells? See that helper cell and killer cells and memory cells. So these are the 3 cells also in between the which one, you know, macrophages, cell, dead cells. or affected infected cell or other cells. Okay, in between there is a mediator cells also participate. That's why it is called a cell mediated. In between some cells are participate for the killing of infected cells. Okay, that's why it is called as what is cell mediated immune system. Okay, helper cells here. This activated helper cells stimulate the killer T cell and B cells, and these killer T and B cells start dividing and produce a clone group of similar cells. This phenomenon is called as clonal selections. Here, what happened? This process is called as clonal selections. Two cells are involved, the helper cell and T cells, both are involved. As a result, the production of mitosis, that is called as what is that? Clonal process. This process is called as clonals. Exact copy of the which one? Memory cells produce a number of cells. It act upon the infected cells and finally kill the or cell dead. So this is called as which one? See here, that things. This phenomenon is called as clonal selections. They produce, this is very, very important. The clones is very, very important. Okay, they produce lymphokinins, messenger molecules, which cause accumulation of WBC to the affected site. Sometimes what happen, this is directly involved with two cells, isn't it? Sometimes what happen, any other blood components, which one in WBC? What are the WBCs? Neutrophils, basophil, eosinophil, lymphocytes, monocytes. They are all are considered getting. They also divide into two types: granulocytes. The cytoplasm, suppose if it is possesses granules, they are all are considered getting known as granulocytes. Monocytes, lymphocytes, they are all agranulocytes. The cytoplasm do not have the which one granules. Okay, that WBC accumulate on it. As a result, what happened? The production of lymphokinase. Lymphokinase is also one of the which one antibody properties. And then again, what is it? T helper cells also stimulate B cells to produce antibodies. Here, T cell and B cell, thymus cell and bone marrow cells. Here, WBC also. Okay, these are helper cells. Okay, and T helper cells also. That means thymus helper cells also stimulate B cell to produce the antibodies and facilitate the action of other T cells also. Not in the T helper cell. And also thymus cells also. Okay, they can induce by the. It is also help to induce the cells. That's the reason. Helper. Okay, so these are all very, very important things. And killer T cells. These cells are clone or these cells are destroy the infected cell or target cell and kill the pathogens. Suppose it will not directly kill the pathogen. Just imagine. It only infected, act upon the infected cell or target cell only, and automatically the target cell is destroyed, means the pathogen it will not survive. That is the reason. And also the cancerous cells by secreting the lymphotoxic substances and secrete the lymphokinin, which attract the phagocytosis, otherwise called a cell eating process. When the lymphokinin secretes, okay, no number of that. What is the meaning of lymphokinins? Lymphokinins, lymphatic tissues secrete the protein molecules. Okay, it has the capacity to phagocytes. Phagocytes means what is that? Cell eating process. So as a result, there are so many cells present in your body. Each and every cells during the infectious stage. Suppose it is affected by the foreign particles, automatically lymphokinin secretion takes place. Otherwise called as cell secretions. Okay, see, these are responsible for the cell mediated immunity. They also destroy transplanted and other foreign cells also. Destroy the transplanted. Which one is the transplanted? Which one is invaded? Other foreign cells also. What are the foreign particles? It means the virus, protozoans, and also bacteria, and also cancer cells, so whatever it may be, that can be easily destroyed by this method. Killer cells, killer T cells. See, and then third one is called a suppressor cells. The suppressor cells also denoted T as. These suppressors, the functions of T cells, thymus cells, T helper cells, and B cells and plasma cells also affected by TS. Cells by synthesizing suppressor factors, because these are the cells, isn't it? It is helped for the production of T cell, B cell is okay. Suppose if it is not produced like that, what will happen? the suppressor cells, what happened? It can produce one of the factor is called a suppressor factor. The suppressor factor activate upon the T cell and B cell and the plasma cells. What happened? Automatically it can be affected by the suppressor cell. It cannot produced. Okay, it is a non-beneficial effect given by the suppressor cell. Suppose if it is production of over, what happened? Over cell production is the beta cells. It can be suppressed by automatically suppressor cell activated. As a result, beta cells and plasma cells automatically affected by these cells due to the synthesis of suppressor factors. And then last one is called as memory T cells. They do not kill the pathogen. Already I told you the memory cells, memory T cells or memory timer cells, they will not kill the pathogen or do not form any antibodies, but these cells retain the memory of every encounter and suppress the entire immune system for attacking the own body. Okay, this is also called as memory T cells. They will not produce any antibodies, but at the same time, what happened? It is attacking the own body. They convert into effector cells on later encounter with specific antigen even after several years. They convert into effector cell. Which cell? The memory T cells is also converts into effector cells. Then what happened? It encountered with specific antigen even after several years. That's why we are calling which one? It is called as memory T cells because it's present in our body, but it do not kill any pathogen. It do not kill any pathogens, but it induce or convert the effector cells. What is that? Convert the effector cells. The effector cells encounter the which one? Foreign particles. It may survive so many years. That is the reason for memory T cell. This is called as immunological memory also. And then antigen presenting cell is also present in it. In immune mechanism, every antigens molecule is processed by antigen presenting cells like macrophages or beta lymphocytes. Antigen presenting cells, what is the meaning of antigen presenting cell? Any one of the macrophages, take an examples of a WBC. Okay, they are having one specific antigen on their surface. Okay, B lymphocytes also, it has a specific antigens. Okay, that's why antibody react with the antigens. So the antigen antibody complex formation takes place. That condition is called as agglutinogen. Okay, this process antigen is presented on the surface of these cells. That's why it is called as antigen presenting cells. Those who are all presenting the antigen on their surface, they are all called as antigen presenting cells. The antigen presenting cells, example, macrophages and beta lymphocytes. When a T helper cell lymphocyte passes closely by the side of the antigen presenting cell, bearing the antigen on its surface, it recognizes the antigen and become activated. When the T helper cells lymphocytes passes closely, so if we find out the pair which is responsible, which is a pair of me, like that, he find out the which one closely, when it passes closely by the side of the antigen presenting cell bearing the antigen on its surface, it recognizes the antigen and become activated. It is for me. So it activated directly, it bind with the antibody and activated. Now T helper cells activate the B cell and T killer cells also. These cells in turn develop clone by frequent division in themselves. Once the T cells activate, T helper cell activate into the B cells. Already we studied, isn't it? When it near to the passage closely, the antigen identification of the T helper cell, it bind with the B cell antigen. As a result, the B cell produce the T killer cells. Okay, automatic the clone process also started. Okay, this is the important progress. See, antigen presenting cells. This is the normal cell, isn't it? Okay, see here there is an antigen is there. See here, when it passes this way, what happens? Automatically bind with the, so which cell it has a antigen. Okay, that is called as antigen presenting cells. This is also another macrophages. Also, it has an antigen. binding sites. Okay, and also beta cells also has a antigen binding cells. Okay, suppose if it is near any one of the foreign particles comes out, so the antibodies, matched antibodies comes out, it may be bind with this one. Here the antibody is there, isn't it? When the corresponding antigen comes here, it also bind with the Fab region. Fab is nothing but the antigenic binding site. This is mostly present the variable region. We will discuss it in the antigen antibody structures. And then second one is called as AMIS, antibody mediated immune system or humoral immune system. This immune system is based on the B lymphocytes 10 to 20%, and these B lymphocytes secrete the antibody. Last class, what we discussed was last slide. What we discussed? Cell mediated immunity, CMIS. Now we discussed which one AMIS. Know the difference? Cell mediated immunity system, antibody mediated immunity system. That one is called as 80%. This one is called as 20%. Okay, note it down. This is very, very important. The antibody or immunoglobulin, that is called as antibody mediated immune system, is nothing but that it is also related with immunoglobulins. The immunoglobulin, otherwise called as antibody. So antibodies are third line of defense. Once again, repeat for first line, second line, third line of defense. They are proteinous substances present in the blood plasma. Okay, these are complex glycoprotein molecules produced by the B lymphocytes in response to an antigen. It is also called as agglutinin. Already we discussed it. Antigen antibody reacted. What happened at the clumps formation takes place. That process is called as anti-agglutination. Okay, the corresponding respective antigen antibody reaction is called as agglutinations. Okay, here you must note it down. This is also so many times they were asked the immunoglobulins. The protein, it has a resistant power. Which one of the following, like that they may ask the question. So immunoglobulin. The antibody is made up of glycoprotein molecules. The immunoglobulin is made up of a glycoprotein molecule, which is secreted by the beta lymphocytes. So, okay, the antigen antibody reacted as a result of formation of agglutinin. Okay, like that, you should note it down. This is called as antibody structure, otherwise called as immunoglobulin structures. Here we should know there are two type of chain is there. One is called as light chain, another one is called as which one? Heavy chain. This one is called as light chain, this one is called as heavy chain. Okay, the light chain is also called as kappa chain or lambda chain. Okay, kappa chain, K denoted as K and also L. So this is the area for which one antigen binding site, both are side present in it because it is a immunoglobulin A or G. A. Heavy chain, see here, immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin D, immunoglobulin E. These are all depends upon the air structure of molecular weight. Here there is a bond. See here there is a black color structure is there, isn't it? Black color bond is that. This is called as bond between the two chain. Sulfhydryl SH. Here also the 4. These are the things. You must note it down. This is the antigen binding site. So particularly the immunoglobulin, it has a 2 chain. One is called as light chain. Another one is called as heavy chain. The light chain is called as, this is the outermost. This is the light chain. This one is called as heavy chain. Each one it has a which one molecular weight. We will discuss one by one. Up to this level, we will finish it like this. Each antibody is made up of 4 polypeptide chain. 2 long chain is called as heavy chain. Okay, 2 short chain is called as which one short chain, isn't it? Long chain and also short chain. Each of the molecular weight 50,000 Dalton, and short chain is called as light chain. Each of molecular weight is 25,000. 50% of the 50, isn't it? Heavy chain 50,000 Dalton, and then light chain 25,000 Dalton. Okay, so hence it is represented heavy chain to light chain 2, heavy chain to light chain 2. These two chains held together by disulfide bond in shape of Y molecule. Suppose the presence of, they look like a Y shape because the presence of the disulfide bond. They may ask the questions why that, how it will be look like a Y shaped in antibody or immunoglobulins due to the presence of the which bond, disulfide bond, covalent bond, hydrogen bond, like that they may ask the questions. Disulfide bond is the correct answer, isn't it? See that? And we will finish it up to this level. Two top tips of these bind with the antigen, the large and complex foreign molecules, mainly protein, that activate the specific immunity, like lock and key model. The particular lock can open the particular key, isn't it? So likewise, the particular antibodies react with the specific antigen only. Okay, phasons and they make antigen antibody complex. As a result, what happened, antigen antibody complex, similar to that of the enzyme, when it reacts with the specific substrate, it gives enzyme substrate complexes, isn't it? Likewise, the antigen antibody complex also produced here. The heavy chain is also called as H chain, whereas the light chain is also called as L chain, which may be either lambda or kappa type. L is denoted as lambda, K is denoted as kappa. We will discuss that things and the next classes. Okay, today what are the things we will discuss? Will you please? So what is by immunity and what are the types of immunity? Active and passive. Also further divided into natural and artificial immunity also is there, isn't it? So, and what is meant by cell mediated immunity? What are the types of cell mediated immunity? What are the cell is responsible for the immunological process? Already we discussed that things is called as helper cells, okay, killer cells and suppressor cells, memory T cells, and also which one the antigen presenting cell, isn't it? And then last what we discussed, what is my immunoglobulin antibodies, okay, and also AMIS, okay. So antibodies, immunoglobulin. What is the meaning of immunoglobulin? What is the types of immunoglobulin? We will see the remaining things. What are the main structure of the immunoglobulin? Already we saw it, isn't it? So remaining things we will see on the next classes. Thank you so much, my dear students. If you have any doubt, please ask me. You have any doubt in this area? Ajay, Ritika, Dhanusri. Nothing else? Hello. Ajay, you hear my voice?
Yes, sir, you're audible.
Okay. So that things already we discussed. Okay. So today we will finish it up to this level only. We'll see the remaining on the next classes. Okay. So what are the things we are going to see the remaining classes? Immunoglobulin structures, and what are the types of immunoglobulin? How many percentage present in it, and also their structures, and what is by G, what is by A, what is by M, and also E, and also D, like that we will see in the next classes. Each one it has a specific features also. Okay, that things we will see in the next class. Okay, bye. Thank you, ma'am. Thank you so much.
Thank you, sir.
Thank you, ma'am.
Thank you, sir.
Thank you, ma'am. Thank you.